And let's move to another speaker, to Sahar Moghimi. She also unfortunately was not able to come to Brisbane, but she recorded a video of her talk. So welcome, Sahar.
Hello everyone, my name is Sahar Moghimi, and I'm very sorry that I cannot be there with you today for professional reasons, but I will be presenting to you in a story our recent works on the characterization of spontaneous activity and neural response of premature neonates to exogenous stimulations during the third trimester of gestation. So to give you a background before going into our studies, if we consider the neurodevelopmental phenomena and mechanisms, and we ask the question, so what is guiding these mechanisms? We know from different studies that these phenomena are guided next to the neurogenetic events, of course, at the beginning by the endogenous spontaneous activity in the brain, and then later on also by the information that arrives to the brain from the external world. So if we consider the period between 24 weeks gestational age and birth, this corresponds to the period that we record EEG from premature newborns in the NICU. We see that there is an overlap, and both endogenous and exogenous dimensions are impacting the development of neural circuits. So it is reasonable to consider that for better characterization of early neurodevelopment, it is important to consider both dimensions, endogenous and exogenous, to develop efficient biomarkers to evaluate early neurodevelopment. And so that's what we've been doing. And I will start by presenting our work on characterization of spontaneous neural activity. And here, what you see is a synopsis of cerebral maturation based on electrophysiological data, so created by Fabrice Valois. And here we see different neurobiomarkers, so specific events at the top in red that appear, disappear, appear, change in characteristics, and then disappear. And then at the bottom, marked by blue, we see the evolution of the characteristics of the background, let's say, bursts of activity. So we are particularly interested in one of these biomarkers, theta activity in coalescence with slow waves. That is the earliest neurobiomarker recorded notably over the temporal regions in premature infants. So we have an example of this event here in high-density EEG. And what is interesting is that there is, not only in this event, but also in other neurobiomarkers, precise phase amplitude coupling, as we've shown in one of our studies, of fast oscillations by the slow wave. And we consider this phase amplitude coupling to be a generic mechanism of early spontaneous oscillation. But what is important is that in a cross-sectional study between 25 and 32 weeks gestational age, when we looked more precisely at this event at its source over the temporal regions that was extracted from high-density EEG, we found that the phase of coupling evolves with increasing gestational age in a way that neurodevelopment leads to systematic temporal accumulation of the theta activity towards the trough of the slow wave. So not only we have this generic mechanism, but also it shows a systematic neurodevelopmental trend. Then if we consider the bursts of oscillation, so no particular neurobiomarker, no particular oscillation, but the general pattern of the bursts of activity that is a characteristic of this age, so the periods of burst, interburst, burst, interburst intervals that with the duration that evolves also with neurodevelopment. Again, in another cross-sectional study, this is a different population, so older a bit, so between 28 and 37 weeks gestational age. We saw that neurodevelopment leads to the evolution of the 1 over f exponent of the aperiodic component. So we are not addressing here oscillations, but the the aperiodic characteristics of the spectra of EEG data, in a way that the 1 over f exponent decreases with increasing gestational age, at least until the age of term, with the appearance of high frequencies over the bursts of activity. So if we consider the other dimension, so the exogenously induced neural activity, so until now we were on the spontaneous activity, but what if we stimulate the brain during this period, notably here because we work on auditory protocols? It is interesting to consider that the brain processes this type of information very early on, because we know that the auditory system is functional probably from 24-25 weeks gestational age. So, if we consider the thalamocortical loops, we know that the thalamic afferents arrive at the cortical plate probably from the beginning of the 3rd trimester of gestation. So the cortex becomes directly connected to the outside world from the beginning of the 3rd trimester of gestation. In a cross-sectional study, we simply evaluated the neural response to single auditory stimulation, so single auditory tones. And what we noted is that with increasing gestational age at birth, the spatio-temporal complexity, so not only how the temporal, how the amplitude evolves in time after stimulation, but also how distributed cortical regions are involved in the processing of this information. So the spatio-temporal complexity increases with increasing gestational age at birth, at least again until the age of term. And what is interesting is that if we put these information together, so meaning here the 2 dimensions, the exogenous and the endogenous dimensions together, so not only how the bursts of activity, as I showed you, characterized by the 1 over f exponent evolves, but also how the brain responds to single tone stimulations, we arrive at a better characterization of cerebral maturation, showing the importance of considering both endogenous and exogenous dimensions together for evaluation of neurodevelopment. But we know that the brain is exposed to information coming from, like I mentioned, from the to the information coming from the outside world. But the development of neural circuits and also networks allow more sophisticated encoding of auditory information, and we know from different studies, notably animal studies, that not only the characteristics of the fundamental characteristics of sound and acoustic information, but also the regularities in auditory streams, like temporal or statistical regularities, play an important role in the development of neural circuits. So we know also that, considering fetal development, that the fetus, so before the age of term, is exposed to these regularities. So meaning, for example, if you consider the maternal sounds, the fetal sounds, maternal heartbeat, respiration, and also the filtered version of the sound coming from the outside world, we know that this information is also, these regularities are also present even before the age of term. So that's why since a few years ago, so 2022, we have this project that started in our laboratory to evaluate the development of auditory rhythm processing during the 3rd trimester of gestation, and also the impact of early musical intervention. So this is a study, this is a longitudinal study where we characterize the early capacities for auditory rhythm processing, and also the follow-up of infants receiving the musical interventions with different protocols. If you're interested, you can check out some of our studies listed below. But today I will precisely show you one of these cross-sectional studies that we conducted to evaluate the development of neural synchronization to different rhythmic periodicities. So this is a cross-sectional study, again, between 28 and 37 weeks gestational age. And the results that I will show you is based on a simple rhythm, so a 6-bit rhythm. As you see, and you have the spectrum at the bottom, so we have periodicities that correspond to the basic level, so the beat, and then the grouping of beats by 2, so the duple meter, and by 3, the triple meter. To evaluate the neural response synchronization to this auditory sequence, so in order to be less impacted by the SNR of the signal and to be concentrated on our frequencies of interest, we developed a phase synchronization index, so a measure, where we model at each periodicity, at its frequency, the periodicity by a sinusoidal, for example, for the beat, and then we look how well the narrowband EEG signal at the same frequency is aligned to the sinusoidal. So this is a complex index. The amplitude shows the how well the strength of coupling, and the angle shows the phase delay between the two signals. So what we have here is, so the x-axis is the gestational age at birth, and what we see here is that with increasing gestational age at birth, coupling strength increases not only at the beat, so the basic periodicity, but also at the grouping of the beats. And this is not related to the, as you see here, it is not related to the evolution of the background activity, but it is specific to the response to the stimulation. And interestingly, if we consider the phase, here we see that neural synchronization here, what I show you is for the beat, so the basic periodicity goes from high variability at the population level to a stable phase with decreasing phase differences between the stimulation and the neural response. So neural synchronization to periodicities at different metrical hierarchies improve with increasing gestational age at birth during the 3rd trimester of gestation. So I will finish my presentation by showing some very preliminary results on the development of OPM MEG in Amiens. So this is a project that is still ongoing, but we started recording with the OPM MEG in children, in epilepsy notably, in adults for different protocols, of course, and recently in neonates and premature neonates, and we are working on the development of fetal MEG. So, for instance, just to show you examples, what we have here is spontaneous, so there is no stimulation, spontaneous activity in a premature infant at 36 weeks gestational age. So what you see here is that we have some rapid oscillations, signatures of neural activity at this gestational age. So that shows that even that using OPM MEG, we get to capture these spontaneous signatures of neural activity corresponding to the developmental stage. And then what we have here is a protocol that started very recently in our lab to characterize the neural response to rhythmic sequences with the objective of evaluating beat perception in neonates. This is a study that is going on in adults, but also in late premature newborns. So at the top, we have the spectrum of the stimulus. So we have the frequencies that correspond to different periodicities, but what is interesting is that listening to this rhythm, I will not run examples because I'm not sure that it will work, but in the presentation, But what is interesting, based on previous studies, listening to these rhythms lead to the perception of a beat frequency at 2 Hertz, so shown with orange arrows, that is not present in the acoustic signal, but emerges during the neural response and also in behavioral responses. So we see these frequencies very clearly in the adult MEG data. So we have the superposition of all the channels, some very preliminary results, but also we have them in premature infants, a bit less pronounced, but still there. So which is very interesting. So we need to dig deeper. We need to record more data to dig deeper in these neural responses.
Okay, let's thank Sahar for her amazing talk. I think that's it. And let's move.
So to sum up, on characterization of spontaneous neural activity, I showed you that phase amplitude coupling between fast and slow oscillations evolves systematically with increasing gestational age at birth, and also that if we consider the bursts of activity, that the linear slope of the EEG aperiodic component also changes systematically and decreases with increasing gestational age, at least until the age of term. On the exogenous dimension, I showed you. Thank you very much for your attention. So I would like to acknowledge my team, our collaborators, and also our funding agencies. Please, because we are not there to discuss with you, please do not hesitate to write to us if you have any questions or comments on the studies. And of course, I will show you, like just mention a short, very short advertisement. So we have postdoc positions available. If you're interested, please do not hesitate to contact us. Thank you very much and have a nice day.